Welcome to the Lundbeck Foundation and to this event where we're about to announce the 2017 Brain Prize. My name is Kim Krogsgaard and I'm the Managing Director of the Brain Prize here at Lundbeck Foundation. First speaker this afternoon is Professor Gunhild Waldemar, who will announce the winners of the Brain Prize. Gunhild is member of the Board of Trustees at the Lundbeck Foundation and chair of the Grant and Prize Committee. So, Gunhild, please come forward.
Ladies and gentlemen, on behalf of the Board of Trustees of the Lundbeck Foundation, I would like to thank you for joining us for this session here today, where we are about to announce the 2017 Brain Prize, the 7th prize in 7 consecutive years. Before the announcement, I would like to extend my deep-felt gratitude to the scientists around the world who have submitted many nominations of very high quality this year as well as in previous years. We are indebted to the eminent members of our selection committee, chaired by Sir Colin Blakemore. Once again, the committee has dealt with a very large number of nominations, and this is not a small accomplishment. Our special thanks go to Sir Colin Blakemore, who is about to reach the end of his term of office as a member and chair of the selection committee. Sir Colin, it has been a pleasure working with you. Your advice and guidance have been of utmost importance in laying the foundation for the Brain Prize, and you can take great pride in the way the prize has grown in visibility and prestige. It's our ambition and goal that the Brain Prize will continue to grow in terms of visibility and impact and become the beacon for international neuroscience. The Board of Trustees of the Lundbeck Foundation has decided that the Brain Prize 2018 and onwards will have a full international profile, and that future prizes are awarded to scientists, regardless of nationality, age or gender, for outstanding contributions to neuroscience, basic to clinical. And now to the announcement. The Brain Prize 2017 is awarded to Peter Dayan, Ray Dolan, and Wolfram Schulz for their multidisciplinary analysis of brain mechanisms that link learning to reward, which has far-reaching implications for the understanding of human behavior, including disorders of decision-making in conditions such as gambling, drug addiction, compulsive behavior, and schizophrenia. Congratulations to you all, and thank you for your attention.
Thank you, Gunhild. Next speaker is Professor Sir Colin Blakemore, who will give us the reasons for the award of the Brain Prize this year. Please, Sir Colin.
Thank you, Kim, and I use this opportunity to thank Gunhild for her kind remarks and to say what a pleasure it's been to work with the Lundbeck Foundation during these early years of the development of the Brain Prize. And let us assume that these are the early years for this very important contribution to the development of neuroscience around the world. The nervous systems of even simple multicellular animals contain circuits of nerve cells, neurons, which are constructed on the basis of inherited mechanisms, enabling animals to make appropriate but stereotyped responses to important signals in the environment. We call these reflexes. For instance, the taste of food or the feeling of liquid in the mouth elicits salivation, chewing and swallowing. Sensory signals exchanged between individuals can trigger mating behavior. The sensory detection of distinctive features of predators can evoke fearful reactions and efforts to escape, even without any previous experience of those predators. So these are all inbuilt reflex reactions that obviously help animals to survive. However, complex animals actively exploring the world, struggling to survive, have to be more flexible and resourceful than a set of simple inherited reflexes permits. Animals need to learn to direct their decisions and their actions towards rewards that will satisfy their needs, or away from indications of danger. This means that they have to learn the predictive characteristics of things and events in their environments. For instance, if you feel hungry and you see a building with a sign that says restaurant on it, you're likely to go inside because the sign predicts that your hunger will be reduced if you do go inside. Early attempts to understand learning in the first half of the 20th century were dominated by a movement called behaviorism. Psychologists, including Watson and Skinner, tested the capacities of animals to solve problems and to distinguish between different sensory cues in order to find food or water, or to avoid some mild punishment, such as a noise or a brief electric shock. They tried to work out the rules by which animals made these decisions. And one conclusion from such studies was that learning is sustained by reward, that is, by the acquisition of something that the animal needs. But the behaviorists generally took a black box view of the brain, unconcerned with how the link between reward and behavior might actually be implemented in neuronal circuitry. In the 1950s, three significant developments set the scene for the research of the winners of the Brain Prize for 2017. First, the sophistication of mathematical approaches to learning increased significantly, especially with the application of probability theory and through growing interest in artificial intelligence. Second, research on brain structures and pathways that might mediate learning was given a huge impetus by the discovery of what's called brain stimulation reward. In 1953, James Olds and Peter Milner showed that rats would readily learn to press a lever to deliver electrical self-stimulation through an electrode implanted in the brain. In some cases, the animals would press the lever at a prodigious rate, apparently without satiation, ignoring food and water, to the point of physical exhaustion, as if the stimulation itself was intensely rewarding. But empty reward like that, produced by self-stimulation, seems similar in many ways to the effects of addictive drugs. And this similarity is more than coincidence. It turns out that both addiction and brain stimulation operate mainly through a small population of neurons that use a particular chemical called dopamine. It was the Swedish Nobel laureate Arvid Carlsson who discovered that dopamine is released from the terminals of the fibers of some nerve cells in the brain in order to transmit signals to other neurons. Although only about 1% of the cells in the brain use dopamine as their chemical transmitter, they play various crucial roles in brain function. Researchers in Lund and Stockholm in Sweden developed methods to visualize nerve cells and their fibers containing dopamine under the microscope. And they found that one bundle of dopamine-containing fibers, the so-called mesocorticolimbic pathway, runs upwards from a structure in the middle of the brain to a small region called the nucleus accumbens, and to the prefrontal cortex and the amygdala, regions thought to be involved in the control of emotion. Evidence grew through the 1970s that this system of dopamine neurons is essential for normal learning on the basis of reward. In 1972, a Swedish researcher, Urban Ungerstedt, reported that selective damage to dopamine neurons with a particular neurotoxin called 6-hydroxydopamine causes deficits in eating and in drinking. And others found that pharmacological blockade of dopamine transmission reduces or eliminates the rewarding quality of food and can extinguish previously learnt habits. Indeed, it became clear that the most powerful rewarding effect of brain stimulation, described by Olds and Milner, might have been caused by stimulation of the dopamine fibers of the mesocorticolimbic pathway. Wolfram Schultz is a German who works at the University of Cambridge in the UK since 2001. He's Professor of Neuroscience and a Wellcome Trust Principal Research Fellow. He studied mathematics, medicine and philosophy at the universities of Hamburg and Heidelberg in Germany, did postdoctoral training in Göttingen in Germany, at the State University of New York in Buffalo, and from 1976 to 77, he worked in the laboratory of Urban Angerstedt at the Karolinska Institute in Stockholm, where he developed an interest in the function of dopamine neurons. In 1977, Schultz moved to the University of Fribourg in Switzerland, where he developed methods for recording from neurons and fibers in dopamine pathways in awake-trained monkeys. Working with colleagues including Ranulfo Romo, he described the responses of presumed dopaminergic cells while the monkey was learning tasks. Initially, the activity of these nerve cells seemed rather puzzling. Before learning, the neurons respond whenever a reward, that is a drop of fruit juice in these experiments, is given to the monkey. If a visual stimulus is then delivered in advance of each drop of juice, the pattern of response changes as the animal learns. The visual stimulus itself starts to evoke a brief burst of impulses from the neuron, and the burst previously associated with delivery of the reward shrinks and gradually disappears when the animals learnt the association. Crucially, if the reward is unexpectedly, to the monkey, unexpectedly not given on some trials, the activity of the dopamine neurons actually decreases immediately after the time when the reward should have occurred. Conversations with Anthony Dickinson, a behavioural psychologist from Cambridge, helped Schultz to relate his findings to current mathematical theories of learning. And a crucial breakthrough was made by the second prize winner, Peter Dayan, when he was working as a postdoctoral fellow in Terry Sinofsky's lab at the Salk Institute in California in the early 90s. Dayan is British. He studied mathematics at the University of Cambridge and did a PhD in artificial intelligence at the University of Edinburgh, focusing on statistical and neural network models of learning. He was a postdoctoral fellow at the Medical Research Council Centre for Brain and Behaviour at Oxford before he went to the Computational Neurobiology Laboratory at the Salk Institute, followed by work at the University of Toronto. He held an assistant professorship at MIT and moved back to the United Kingdom in 1998 to a position at the Gatsby Computational Neuroscience Unit of University College London. He became its director in 2002, and a position that he still holds, as well as a chair of computational neuroscience at University College London. In 1996, Dayan, together with fellow postdoctoral fellow Reid Montague and Terry Sinofsky, published a theoretical paper based largely on Wolfgang Schultz's observations, arguing that the changing pattern of activity of dopamine neurons in monkeys during learning suggests that they are implementing a particular computational procedure. Previously, mathematical psychologists have proposed that learning is driven by deviations or errors between the predicted and the actual magnitude and timing of reward. This reward prediction error theory has been highly influential in computational approaches to learning. And Dayan and his colleagues suggested that it might be implemented by dopamine neurons in the brain. Schultz then collaborated with Dayan and Montague, producing a seminal paper in 1997 in which they showed that the activity of dopamine neurons during learning could be simulated by a computational model incorporating the so-called temporal difference algorithm, assuming that neural circuits can somehow construct a temporal label signifying the time between the stimulus and the predicted reward. Both Diane and Schultz have broadened the scope of their work and made further important contributions to the field of learning and decision-making. Wolfram Schultz continues to study neuronal activity in many parts of the monkey brain, including the striatum, the orbitofrontal cortex, and the amygdala during different forms of learning, risk-taking, and economic decision-making. In a very recent, technically brilliant study, Schultz and colleagues, which includes Edward Boyden, one of the winners of the Brain Prize in 2013, developed methods to introduce light-sensitive molecules selectively into dopamine cells in monkeys, allowing the researchers to add extra impulses into the activity of those neurons while the monkey was learning. They found that such extra activity, added to a real reward, it's a drop of juice, enhanced the value of that reward, making the monkey prefer a visual stimulus that predicts the enhanced reward. The increased value was evident after learning in the stronger responses of dopamine neurons to the visual stimulus associated with the fruit juice plus the extra optogenetically induced activity. This provides strong evidence that the prediction error signal conveyed by dopamine neurons is actually causally involved in the learning process. Schultz has also extended his research to behavioral and neuroimaging studies in humans. He's used neuroimaging in collaboration with Nico Linders to study activity in the human brain related to monetary reward, changes in reward-related signals in Parkinson's patients, in smokers, and in individuals with opiate addiction. And his most recent work has focused on neuroeconomic studies, including such subjects as reward value coding, the influence of risk on reward value, the neuronal coding of economic utility, reward, and decision signals. Diane is recognized internationally as a leader in the rapidly developing field of computational neuroscience. He's also pursued an extensive and productive collaboration with the third prize winner, Ray Dolan, on the issue of learning mechanisms, including reward prediction error, using behavioral methods and neuroimaging in humans. Ray Dolan is a psychiatrist who has been a leader in the development and the exploitation of human neuroimaging. His productive collaboration with Peter Dayan has produced a total of 47 jointly authored papers. Their initial study, published in the journal Neuron in 2003, was one of the first to use fMRI to link Schultz's observations in monkeys to learning in humans. They used fMRI and regression analysis to reveal regions of the brain that show a shift in the timing of activity from presentation of the reward to presentation of the conditioning stimulus during appetitive conditioning, using a pleasant taste as the reward. And they found such a shift comparable to that seen at the level of individual dopamine neurons in monkeys by Schultz in the ventral striatum, the region of the nucleus accumbens, and in the orbitofrontal cortex, which are both targets of the dopamine projection from the midbrain. Dolan and Diane's collaborative work has ranged over many aspects of learning and decision-making, and one highlight is a recent study reported in Nature Neuroscience in 2013, in which they showed a reduction in older, healthy individuals compared with young adults in activity of the nucleus accumbens that was produced by reward. They also used an imaging technique called diffuser tensor imaging to assess structural connectivity between the midbrain and the nucleus accumbens, and found that the degree of connectivity in each individual person was correlated with the level of the response in the nucleus accumbens related to reward prediction error. And moreover, they found that administering the dopamine precursor chemical L-DOPA, used in the treatment of Parkinson's disease, they could restore the learning potential in some individuals, and that this correlates with enhancement of the prediction error signal in the nucleus accumbens. Diane and Dolan have worked on such diverse extensions of learning theory as a computational and neural modeling of well-being, dopamine modulation of decision-making and well-being, striatal structure and function, correlated with individual differences in pain avoidance learning, the effect of dopamine on value-independent gambling propensity, and the influence of brain stimulation in Parkinson's patients on the sensitivity to decision outcome. And in a recent collaboration with Trevor Robbins and other colleagues in Cambridge, Diane has extended his interests in the clinical implications of work on learning theory with a study of learning habits in obsessive-compulsive disorder. They've shown that a bias to habit-based decisions as opposed to goal-directed decisions correlates with lower grey matter volume in parts of the brain. The Brain Prize then, for 2017, recognises a remarkably broad multidisciplinary analysis of brain mechanisms that link reward and learning, and it has far-reaching implications for understanding human behaviour, particularly disorders of decision-making in conditions such as gambling, drug addiction, compulsive behaviour, and schizophrenia. Thank you.
Thank you very much, Sir Colin. The last speaker today is Professor Morten Kringelbach, Aarhus University and University of Oxford, who will put this year's prize award into a Danish neuroscience context. So please, Morten.
Thank you so much. And thank you, Colin, for that erudite exposition of the work. I think it's safe to say that these are truly exciting times for neuroscience, as we're gaining more insights into the fundamental brain mechanisms. It's also very clear that reward is really central to understanding human nature. In the words of the Nobel Prize winner John Steinbeck, who wrote a book in 1941 called Log from the Sea of Cortez, he wrote, "Man might be described fairly adequately, if simply, as a two-legged paradox. He has never become accustomed to the tragic miracle of consciousness. Perhaps, as been suggested, his species has not gelled, but is still in a state of becoming, bound by his physical memories of a past of struggle to survival, limited in his futures by the uneasiness of thought and consciousness." So why is that relevant? Somebody writing about human nature for so long ago. But really, I think it's safe to say that human nature really is set between exploitation and exploration. And as we've heard today, the pioneering work today is honoring the finding that dopamine is very much involved in prediction and prediction error. In fact, in many ways, we could say that humans are, in fact, prediction machines. In order to survive, we need to pay attention when something surprising happens. In other words, we have to minimize the prediction error. We have to be motivated to pursue rewards and learn from that process. And to take that even broader, think about the pleasure cycles that we go through every day. Right now, I'm in need of coffee. I'm wanting coffee. I'm trying to predict where my next cup of coffee might come from. When I engage with the coffee, when I have the coffee, I like it. And after a while, I become satiated. I can't have any more. So really what we are looking at here is trying to understand how these shifts in the brain happens. And not just to simple things like foods or coffee for that matter, but even something higher order like music. And so, for instance, here in Denmark, we've recently started a center funded by the Danish National Research Foundation called Music in the Brain, where the basic premise is to pursue the idea that music really could be understood in terms of prediction on many levels. We are constantly making predictions at the level of the melody, at the level of rhythm or of harmony. And it's by constantly trying to make sense of those things that we are, in fact, getting some joy in that process. That tension is hugely important. And of course, the contention is that if we could understand that process, we might even be able to develop more effective treatments. Because, of course, music has immense potential to actually deliver exactly that. Think about the way that infants and parents are interacting. Music could be used to calm the child or to get the child to be more active. But again, we need to understand the basic principles, how they operate, not just in humans, but also in those who have not yet got language like infants. So I think that is really one of the exciting things about this year's prize. This possibility that we could learn how to rebalance the brain. And not just rebalance the brain when it comes to motivation, or to the learning, but actually get to the good life. In the words of Aristotle, who talked about hedonia and eudaimonia, pleasure and the well life lived, really what we are gaining from understanding these things is that we can finally look into new ways of trying to rebalance the brain when it becomes unbalanced. And one of the exciting things about Lundbeck Foundation giving out this prize to an international set of scientists is, of course, that a lot of these things are also happening in Denmark. Denmark has become increasingly more international as the years gone by, and I think these last 7 years has been very important for that development. So, coming back then to the sort of the further implications of this, it's quite interesting, I think, if one looks at the American Constitution, for instance, that they talk about the pursuit of happiness rather than happiness itself. Could happiness itself, rather than the pursuit, be something that could finally be within the reach of neuroscience? And of course, we are now seen with the work, the pioneering work that has been honored today, that yes, dopamine plays a role, but at the same time it's also clear that it's linked to a much larger network. And really, there are many questions that remain. What are the larger brain networks that are involved in that are enabling that for phase transition? What are the other neurotransmitters that are involved? So, for instance, the work of Gitte Moos Knudsen here in Denmark has taught us a lot about how serotonin could be a very important part of this. But really what we need is more research. We need to broaden the perspective. We need to use some of the new mathematical tools that have been developed. And really, I think tomorrow's neuroscience is all about making those links. It's fine to make the discoveries, but until we actually have a mechanistic understanding of how those discoveries should be understood, we haven't really understood very much. And so therefore, I think it's highly significant that today's prize honors not just a finding, but an actually understanding of what the mechanistic underlying principles are. Because, of course, once we understand those, we are in a much better position to actually try to rebalance those networks, rebalance those brains, to not only increase hedonia, pleasure, reward, but also eudaimonia, the life well lived. So, thank you very much.